concerns or queries you may ask. So we covered this part. I'm sorry, he's gone on the other screen. So let me swap my, or maybe I can go with this slideshow and custom slideshow, and then I can customize that. Where where is that option? So it's already in slideshow mode, so I guess he's not showing me those options, but who is waiting in the lobby? Alright, so Ahmed Haik. So custom slideshow, why it's not showing? Oh, setup slideshow. So chaotic or individual window screen. It's chaotic. Yeah, so either we can go this way. Save. I'm sorry, I just closed that thing. So let me open that one more time. So we cover some background and then we do some practice, some fun stuff in Linux. Right, so we were here on this data analysis part, and let me check how do my slides they look. Classic full screen and individual window. Right, so no, not this one. Classic. Yeah, so I guess that's what I'm looking for, but it's there on the other screen. So let me share the other screen. All right, so you okay, slideshow and gee, should we install this for Windows or Linux? Yeah, that's issue is there. I could not install that thing myself, so maybe we can fix that and send you the um, maybe send you as an attachment or in a compressed file, and then you can simply place it in your folder, right? So let me share this screen one more time, one last time. So you can go here, stop sharing and share again. And all right, so we are here. Stop display. Yes, this one is just fine. Okay, so I hope that comes here. And now we are looking into different applications. So how we will perform the genome assemblies and how we will perform the de novo sequencing and resequencing, like um, getting the SNPs and CNVs. Right, so we will talk about the exomes and we'll talk about the transcriptomes. The RNA sequencing are shortly called as RNA-seq. Right, so I guess covered all this stuff. So here, this data analysis, so we have that sample, we extract the DNA, we do shotgun, and we attach adapters, and in NGS or second generation we amplify that, but in third generation we do that. In the end, there are some images that are created and computer reads them, and this how it gives you the output. That's a cartoon picture, so you have the arrows going forward and backward. So these are your paired end reads. If you are seeing, these reads in pairs, single reads will be shown in only forward direction, arrows pointing left to right. So that's the single end sequencing. In single end, you can get each and everything. You can break your DNA into short fragments, and you can take all those fragments. Here, you take the ends of some longer fragments, like you take the sample DNA and you can take only its reverse and forward ends, like few bases from the left side and few bases from the right side. So in this way we can just capture their ends. So that's why this technology is called as paired end sequencing. Down there, we already talked about FASTQ format. So the data at this stage is in FASTQ format, but you get the reads from sequencers, but that are also having the adapters attached to them. So you need to remove those adapters. So that step is called as QA or QC, quality assurance or quality control. So once those adapters are removed, or once those reads their qualities and their scores and their base composition or GC content, everything is okay, they are called as clean reads, and those clean reads are then put together, and in this way we can assemble the original genome or original DNA fragments from these individual reads. This thing is called as assembly. Remember, we took the original DNA and then we break it into those small fragments. So at that time, that thing is disassembly. So now we are doing assembly, and once you assemble them and you let it go through various QC stages, this thing becomes a reference. But in most of the cases, the assembly projects are where we are not creating the references. Maybe we can get these small fragments put together into bit larger fragments, and then if the reference already exists, or even the raw reads, if you just take them, you can map them or align them to the reference, right? So in this way, here in this diagram we see that back arrow or back thick line is your reference, and these are the individual reads which are aligned to it. So one of the pair is here and one of the pair is here, forward, reverse, and this is just reverse, and its forward strand may be somewhere here. These are two just forward strands. So this how those alignments are done, and the format is called as BAM format. BAM format. While you have the reference, then what you do is you start doing what we call it as annotations. So in annotations, you find the genes and the coding regions and the exons and the introns and the non-coding regions. So in this way, you are actually finding different regions or different important things into that reference which you built. This thing is called as annotation, and the file which you get is in this format called as GFF. GFF stands for graphic file format. So today we are doing all that stuff on Linux, right? So, and then we move on to this where you have that raw BAM file, that is just an aligned file or alignment file, which you get after you do this alignment or mapping, and then you remove the duplicates. The reads which are starting from same point or ending at same point, they are duplicates. So you remove them because in snip calling those duplicates might do the issues. Still up till here this thing is in BAM format, but when you do snip calling or when you do variant calling, that thing is called as VCF. VCF stands for variant calling format. right? In differential expression, normally we talk about gene expression here. That is where we are doing RNA sequencing or RNA-seq. So over there we let these reads, these aligned reads, we count them, how many reads are aligning to which transcript, and once that file comes in, we process it with the tools like R or Python, mostly in R, and then we do those differential gene expression calls on it, like which genes are differentially expressed in which samples, right? So for example, you have the treated um, patients versus the not treated ones, or the ones you have the control versus the, right? So controls versus the treated group. So in that case, you are performing the differential gene expression. That is where we will spend at least 1 week. So 2 lectures may be needed for that. Okay. Base calling we already covered, that which base is this. Q score is what is the quality of that base, and we covered this thing that originally in Human Genome Project, when they came up with this automated Sanger sequencing, those sequencers were generating the data in the shape of those colored peaks, right? We call it as electropherograms or chromatograms, and then these are processed, and these peaks are then looked for two things. Number one is the sharpness of the peak, uh, or the uniqueness of the peak. So you have some background noise going down, but at some places that noise rises high. So some of the bases are calling that it's T here, but most of them are calling that it's a G here. But obviously that lowers the quality versus the regions where you have less noise. So only G is pointed out here. So that is of high quality. There is no background noise here. Here, this region is noisy. So that means the scores are low in that. So we check that. Here we talked about the FASTA format, and how why we needed FASTQ format, because FASTA was there to store the sequence, and then there was the another file qual for each sample. We were getting two files, and that was there in 454. One where we have the sequence or base calls in FASTA format, whereas the quality scores in another file named as qual. So FASTA and qual, two separate files, but obviously you are having huge sized files, and you need to keep track of these files in pairs. So that's why they came up with this new format, that is FASTQ. So in FASTQ, since we wanted to have one-to-one correspondence between, this nucleotide G has a score of this small letter G, but behind that we actually saw that this small letter G is then translated back into its regional ASCII value, because any key you click on your laptop, it invokes an ASCII value, a number, and that number is then converted into the binary digits, right? So for example, the 32, that number is associated with space character. So when you invoke a space character, a number 32 is invoked, and this number is then translated into this binary string, where each of those bits are in powers of 2. So here we start 1 2 3 4 5. So and then this 6. So first one is we don't count, that's called as the flag bit, but 2 power 5 makes it 32, right? So in this way, that string actually of those binary digits is actually translating this thing into a space character. So those quality scores are then, rather than putting them in numbers, we convert them into those ASCII characters, right? So these ASCII characters are then placed there, and then we also talked about these formulas, how that score is calculated. So Q score or qual score is equal to negative 10 log to the base 10 of P, and here we saw that Q can be converted to P, and P can, and then we talked about the quality score. So score of 10 means 1 in 10 is the is the error rate or the probability of incorrect base. P here is the probability of error. So if error is 1 in 10, that's 0.1 or 10%. Obviously accuracy is 90%, but 20 and beyond is good. So 20 is 1 in 100. The accuracy is 99%. 30 is excellent, and 40 is the best or maximum which someone can see in this NGS data. Here we talked about the Solexa and Fred quality scores, and in Sanger they went with the Fred formula. Solexa came up with the another formula, that's where he was using the ratio between P and Q. So that thing is called as odd. What is the odd? Odds of being smoker versus non-smoker, right? So and then that ratio between getting something versus not getting something is called as odd. So if you are comparing two odds, in that case this thing becomes odds ratio, right? So you might have heard of odd ratios, or then sometimes since the number is very small or in fractions, so they convert it into log scale. So that's why the term log odd is also used. But I'm not covering that thing over here. I just want to tell you that these are the ASCII characters which actually they picked, and each character has an associated ASCII value. So here we added what we call it as offset. Why we need to add the offsets? Because the scores from 0 to 32, you cannot represent them directly with ASCII characters, because 0 to 32 includes special characters. So special characters are the ones which are maybe like space character or enter, that's called as carriage return or tab character. So those are the ones which you cannot see them, but they are there, right? So up from 33 onwards, these are your visible characters. So you can use anything beyond 30, including and beyond 33, right? So 33 has this sign of exclamation. So suppose your actual score is 0. I mean, you get the reads which are all bad, or which just very obviously you will reject them, but a score of 0 is then represented by this sign of exclamation. How? Because it represents 33. So what you need to do is you take that raw score and you add that 33 in it. So that's called as an offset. So Sangers, they go from 33 onwards. Their score, physical score, ranges from 0 to 40. 0 means 100% error, and 40 means 90. What was that? The 90.9% accuracy, right? 1 out of 10,000 fragment copies. The single fragment goes in a sequencer and you get its multiple copies. So suppose if you have its 10,000 copies, only one of them is giving you the wrong indication about which base is this, but rest of them are just fine. So you need to let the people in. Sadhya is having exams, so she's not available. She'll be here shortly. So maybe any one of you can just look into the lobby. You can just let them in. In QC, we can have the issues like, so I think I put it here. So in Solexa came with this formula, this odd scores, but they added an offset of 64 because because of that ratio thing, and you can take the log of a ratio thing. Because of that ratio thing, the minimum possible score was -5. So if you are adding 33 to it, it will become minus 20. It will become 28. So you cannot use the character whose ASCII value is 28. So that they rather decided to add 64 in it. So 64 plus -5 is 59. So they went from here to onwards. Illumina went from. So what they did is a wonderful thing. They took Fred formula from Sanger and offset from Solexa, and obviously their score ranged from 0 to 40. but they added this new offset of 64. So 64 means 0 score, 65 means 1, 66 means 2. So 2 is really bad, so B means bad. C, D, E, and as you go for the higher characters, your score starts becoming good, right? So that's the scoring schemes. What QC issues you can see here, you might get vectors, but normally those cloning vectors are not utilized in NGS. They were utilized in the Human Genome Project for Sanger because Sanger was going slow. Um, so you need to sample the DNA and you need to store it in those cloning vector libraries, but that thing is not here. But sometimes, if you are working uh, with the DNA which you actually stored in cloning vectors, so those cloning vectors may be an issue, but mostly adapters, since you added the adapters, you need to remove them. You can get the reads of low quality or low complexity where your system was sort of confused and it was plugging in ends or anything like where he was not sure which nucleotide is. Contaminants, for example, your sample was there but it got contaminated by bacteria or fungi or anything else. And duplicates, because since you are doing that, though you're taking the fragments and you are doing PCR, so sometimes you will get the duplicates. We talked about box and whisker plots, and this is what it's happening in QC. Here we have the reads, we stack them together, so their sequences are stacked. Their quality scores are also stacked, and then these quality scores are then converted back into their original scores by taking the ASCII value of that letter, for example, this small letter G, and then subtracting that uh, offset from there. You can get the actual scores and you just place them. So you can take these numbers one by one. So all reads are put together, are stacked together like plates, and then you can go into each base position. So number one position, you can go like columns. So in this first column you have G and A and C, so three reads are there. All bases are 33% each. Second position are all T's, and obviously these are their scores. So for the numbers you can go for mean, mod, median, quartile 1, quartile 2. You can plot their box plots, but for those nucleotides you can go for their base composition, like you can go for their frequencies. So how many T's are there, how many G's are there, how many C's are there. So once you do that, FastQC returns us these quality scores or Q scores in the shape of those box plots, and then that shape is divided into three sections or segments. The green zone, all that data which is here is good. Yellow, tolerable. Red, obviously you need to take care of these reads. Boxes represents the midpoint middle class, are obviously the mid 50% of the population. So the lower end of the box is where you have your lower class. So lower class goes from 26 to 36. So minimum possible number is 26, and one fourth, lower one fourth of the population, or lower 25% of the population ranges from 26 to 36. So they are up to 36. Their Q score is up to 36, right? So these are the medians. And so 50% of the population, the midpoints, 50%, that's 39. And this is the Q3 as well as that is the maximum possible score. So then, the top 50% of the population is having a score between 39 and 40. That's excellent. That's how you interpret those box plots. Sometimes their one side is completely missing, but as you see here in those box plots, you have the antennas on this side as well as on that side. So that is here up till your lower quartile or lower 25% of the population, or lower one fourth or lower class. That is where you have your middle class or midpoint. That is your, you can say this is the lower middle from here to here, and this is your upper middle, and this is your upper class. So again, like your 4 class categorization or classification system is here. That's why this thing is called as a quartile plot. Quartile means out of 4. So you take a thing and you do its chunks or segments, or four chunks or 4 segments, having a proportion of 1 by 4 each. Okay. Right. So in genome assemblies, what you do is, in genome sequencing or DNA sequencing, it's a de novo assembly. If your reference is not there, you can take those reads and you can put together on the basis of similarities, on the basis of overlaps. Since you cut them at random, since you get the multiple copies of that startup material when you started this experiment, right? Lower species or smaller species, you might need to do PCRs, but for the eukaryotes or for the individuals who are having, huge amount of DNA in their genome, you can start, you can ensure the copies of the genome or the DNA with your starting material. So how much starting material you are taking, the companies advise you, like if you are taking that microgram or nanogram of the DNA, you can roughly achieve that many copies of your original genome. That thing is called as coverage, right? So that's how your, then once you have the multiple copies, you do their random sharing, you do your random shearing and break them into those short fragments, and then since those fragments are cut at random, we can get those overlapping fragments. On the basis of those overlaps, you can just stack them and you start constructing the bigger chunks from those short reads. In resequencing, you can take your reads and you can align them if a reference already exists. Like most of those human genome projects now we are doing, they are resequencing because we are aligning them into the reference that was provided by Human Genome Project, right? So right now we have HG38, Human Genome 38. That is the assembly which is the latest. So if you sequence your own genome, or if you're sequencing your patient's genome or even exome, you can align them or map them to the reference HG38, right? And then you can see the similarities and the differences. Sterics are the points where you are getting the differences, where there are no sterics. So in real, when you go with these genome browsers, you can actually see what you have on the reference and what you have on those into your sample. So that's actually your sample which you fragmented and sequenced, and then you align it to the reference, right? So that's the complexity of the problem. So that is from a book. This stanza is from the book in Leipzig et al. There was a review from which we have taken that thing in 2010. So they are trying to teach you the assemblies with this example. So that's a stanza written in this book, Charles Dickens. Anybody who's familiar with Charles Dickens? It's these novels. One of them is A Tale of Two Cities, that is famous, and there is a movie on it on YouTube, A Tale of Two Cities. Very beautiful movie. It's about the French Revolution, as you can see in the book title. So they are trying to, cut the heads off or chop the heads of some ministers or some rich people. So there was a big gap between the rich and poor. So on one segment of the society, for them it was the best of times. As the movie starts, you can see that scene where the poor are fighting for food and rich are enjoying their parties and dancing and drinking, and so they say it was the best of times on one side. On the other side, it was the worst of time. So there was so big gap between these two classes that even those poor, they got so much resentment that they started uprising against their government and their officials and their queen. I think who used to said that if they don't have bread, why don't they eat biscuits? So it was the age of wisdom on their side, but it was the age of foolishness. It was the epoch of belief, and it was the epoch of incredulity. So that's just a stanza, a few lines from this, just to teach you that there was this complexity of the problem. So when you put it together in the shape of DNA, this thing goes like this, because DNA is a sequence, right? Here, commas, full stops, quotes, spaces, new line characters, they are in the shape of ADGCs. They are still the characters, right? Do you have these spaces within the genes? Obviously, you have the coding regions and non-coding regions. So, but everything over there, those space characters or those tab characters, they are in the shape of ADGCs. So it looks like this, right? And when you break it into those short fragments, and then you get their sequences, or when you get those reads, they look like this, and your job is to put them together. So that thing is called as assembly. And once you assemble them, obviously you need to get the original message from this. That's the annotation, right? Annotation. So since you disassemble this thing, you need to assemble it back, and then you need to do that annotations. So genome assembly is the process of reconstruction of original genome or chromosomes based on overlaps between short sequence reads. It groups the reads into contigs and contigs to scaffolds. Okay, so reads put together into contigs, contigs put together into scaffolds. Okay, so you start putting the reads together on the basis of the overlaps, and the thing which you get, you call it as contig. And then you take the context and you put together, and you even generate the even longer fragments. So that thing is called as scaffolds, right? And then scaffolds are put together on the basis of some landmarks, right? Some markers. People who are in molecular biology or genetics, they understand that. So, and then you put them into the and assemble them into the chromosomes. So these are the different levels. Originally it was a genome that is broken into the chromosomes, and you took those chromosomes and you break them into those short fragments. But now your job is to assemble those chromosomes back. But these are the stages in which you pass through. When you get from the sequencers, these are reads. When you put them together, context put together are scaffolds, right? So what you do over here is you do sample preps, you do sequencing, you do assembly, and then you finish your assembly. That's a very, very important step, right? So you let that assembly go through multiple QC stages. Finally, you conclude your assembly, right? Contig is nothing but a consensus sequence of DNA that has been assembled from overlapping DNA fragments. Scaffold, one or more contigs linked together by unknown sequence called as a gap, right? So for example, there is this contig number 1, there is this contig number 2, and you know that in between them there are 20 bases or 20 nucleotides that exist, right? But you don't know what you have in here, what those 20 bases are. So that other than that, you just place their seeds or place their spots, and you fill them with n means it can be anything, it can be any. Have you ever downloaded the FASTA sequence of any chromosomes? You will observe that that sequence comes with large number of n, right? N's. And then in the end, the chromosomes also end with the N's. Can you tell me why it is so? So chromosomes ends are having very, very highly variable regions. So you cannot develop a consensus. There can be any nucleotide, and depending upon the species and depending upon the individual, those things are quite different. They are ends. They are called as, anybody in molecular biology or genetics, the ends of the chromosomes, the highly variable
Telomeres.
telomeres, right? And you know that these telomeres, they also contribute to another thing called as aging, right? So they are leading us towards our end, or that's because those telomeres, they are shrinking, their length is reducing day by day, or year by year. And then finally our chromosomes will be exposed, and once they will be exposed, obviously they might be eaten up by the enzymes which are, so we are carrying our death programs also with us. So here you have the reads, those dots are your reads, the blue dots and the red dots. Blue ones are, and obviously these are in pairs. So blue ones have a small insert, a small gap in between. The red ones has the long inserts. So you get a pair of paired end reads. One of the group has the small insert, and one of the group has the large insert. Okay, the small insert reads, the reads that are having, these are the paired end reads. Obviously, you took a segment that was having a length of say 200 bases, and you just captured, say it is yeah, even less than that. Suppose like it's 250 or 300. So you get 100 on this side and 100 on that side, and in between you discarded 100. So that thing which you actually discarded is called as insert. Though this word is quite confusing, because insert is that there is something inserted. So actually there was something that was inserted here, but we deleted that. But the term we use over here is insert. These are the ones that is having the longer insert. So suppose this guy and this guy, the original fragment of was of 1 kilo bases, 1000 bases are base pairs, right? So you took 100 from here, you took 100 from here. How much proportion you did not take or you discarded in this paired end thing? So 100 on that side, 100 on that side. That makes 200. Total was 1000. So how much you discarded? Something like 800, right? So these are the long insert pairs and short insert pairs. So short insert pairs are good to make contigs. Long will help us make scaffolds. So contigs, reads put together into contigs based upon their similarities and differences, right? And these contigs are then placed together on the basis of with that, and obviously that paired end technology helps us here. So you must be wondering why we are doing this paired end thing. If one of the pairs is here, so that means this guy, this contig should be here. If its partner is here, so obviously that means this guy is here. So we can place this contig and we can place that contig. You can also get this length which you did not fill up, so you can pad those gaps here, this one and this one. So this one, for example, is contig 1, which we placed here, then this second, and this guy, the third one we placed here. We know that the distance between these two is 800. So here is this 100 and here is this 100, and this distance between them is 800. Suppose that part is 150 and that part is 100. So what do you have in between? So that's 100 plus 150. So that's 250. 800 minus 250 is, right, 550. So that is the gap which we actually captured. So you need to put those ends here. So in this way you can place those contigs together. The resultant thing which you get, this longer entity, is called as scaffolds. So reads put together into contigs, contigs into scaffolds. Where or which level is acceptable in our assembly project? So obviously in virome or in bacterial genomes, if it's a genome assembly project, so over there, this contig level is sufficient. This contig level is sufficient, and some people they even go at scaffold level. But obviously, for higher organisms like eukaryotes, obviously, to get a proper assembly and chromosomes, obviously you need to go up to chromosomal level, but that makes it complicated or that makes it expensive. Okay. So assembly algorithms, the algorithms are the tools which are built on those algorithms. Algorithms are nothing but steps, right? So those steps over here are put together in the shape of flow charts, or in another thing which looks like a code. We call it as a pseudocode. One of them is all paths, one of them is velvet, and one of them is miraculous. These are just few which we put it here. So we will be using velvet in our assembly example or demo in Linux. I use the word algorithms, so that's a credit to a renowned Muslim mathematician from Iran or Persia. So Persian mathematician, as they have written in the books. He was there 780 to 850, and his book, Algoritmi di Numero Indorum. That's the translation of the original book. The original book was Al-Kharzmi on Indian numerals, Kitab al-Hisab al-Hindi, right? So book's name is Kitab al-Hisab al-Hindi. So Sanskrit has those numerals, so they are then translated into those English digits. So 0, 1, 2, 3, 4, 5, 6. You know, your whole computing or your whole mathematics and your all calculations are very they owe their origin from this conversion of those modern digits. So modern digits 0, 1, 2, 3, 4. How many of them are there? There are 10 of them. Counting zero, zero is very important, right? So zero is just a zero if it is, you know, it's alone. But if you put it on the right side of any of these digits, and obviously they can convert 1 into tens. You put 2, they will convert into hundreds or thousands or 10,000s, and you can go like billions and trillions and whatnot, right? So that was a credit to Al-Khazmi. That is the advantage of doing paired end sequencing. We already covered that, but that in scaffolds you can have this situation. One of the pairs is here, the other one is here, right? So you can place those contigs into scaffolds, and you can also capture the length of those gaps. Coverage is nothing, but it's given by a formula. C is equal to NL divided by G. So total number of reads is N here. Length of a read is L. So if it's, for example, Illumina, who is giving us the read fragments of length 100, and you are getting 1 million of them. So 1 million into 100 is obviously 100 million divided by genome size. So for example, if it is human, divided by 3.2 GB. So how much coverage is that? You can calculate with this formula. So we just practice here. Illumina produced 1 million reads of 100 base pair in length for a species, any species whose genome size is 1 MB, right? So NL divided by G. So N is 1 million, you have 6 zeros in front of 1. See the importance of zeros here. Multiplied by 100, so that's the length of individual reads. So that is the 100 million divided by 1 million, or 1 MB is just 1 million. So 1 million, 1 million cancels. So that species has this coverage of 100x. 100x. What if it's having 0.50 million reads? So obviously it will be 50x. So that is just a simple mathematics. But we know that actually in real life that coverage is not that pretty simple. So it's actually uneven. Some of the regions are highly covered, some of the regions are not good covered. For some of the regions you get good data, you get good fragmentation, right? So and then good representation in your read data, right? So for example, this region got 5 reads aligned to it. So here for example is T, and this is also saying T and T and T and T. So 5 of them are saying T, so that means it's a T here. This region is, for example, covered by 1, 2, 3, so its coverage is 2. This region is by just 2 reads, so that is, coverage is 2. So each base position is then checked for how many reads are mapping to that. So we'll also cover in alignment or mapping part when we do this SNP calling. This region is not at all covered, so that's where you can have maybe deletions in your sample site, okay? Or maybe the regions are having repetitive regions, or some of the regions are quite hard to capture. So obviously those regions are less represented. Normally what happens during an assembly project is that those repeats, they cause trouble. So you have the copy of repeat here, R1 here and R2 here, and in between there is some non-repetitive region. So you have this green part and red part and blue part, so that's normal, but this yellow part is some repeat, and this is its first copy and that is its second copy. So sometimes it happens that all the reads are here and here, they put it together, so that thing comes out. So this thing is called as orphan contigs. So that problem is common if you have highly repetitive regions in your genome or DNA. So there are the tools called as something like repeat maskers or some other tools where you can guess the repeats where are they existing. So those regions you need to be more cautious or more. Again, this paired and read helps us in getting rid of those repeats. So those reads are at random. So for example, first of the pair was here, the second of the pair was here. So that means after this is this guy. And within that, this read pair was here and this was here. So that means this yellow is a piece that should be placed here. After this red, it should not be merged here, right? So that repeat resolution is also you can solve it with the help of those those paired end technology, right? So these are different algorithms, they operate here. One of them is called as OLP, overlap layout consensus. So what you do over here is in this layout thing, you take those reads and you go on the basis of their overlaps, right? So here, for example, this ends with C here, and this is a T here, and this an A here. Maybe this diagram is a little bit screwed up. I think this thing goes here, this thing goes here, this thing goes here, and so on, right? So, and then obviously it's a copy paste thing. Maybe we can rewrite that. All C's here, aligning here, this C aligns here, this T aligns here, and obviously A's, they are aligning here, and then, some of the fragments are going out. So you can take those reads and put together, right? So in this way you need to match every read with every other read. So obviously, since these reads are in millions, so you need to go for, obviously, for the genomes which are larger in size. This thing is impractical. They came up with another thing here, we call it as graph. So they try to solve this thing with graph and graph theory, and and that is the way of resolving that. So each read, we can take it as a dot or as a node, right? So graphs are where individual entities are called as nodes or vertices, and they are connected with arrows, we call them as edges. So you can have r1 here, this one, and then followed by this guy. Even you can take r1 and r3, but obviously that edge is not that strong. And then when you reach here at r4, either you can go this way or you can go that way. So you try to just solve this thing with the help of that graph thing. The most successful approach is called as de Bruijn graph. So in de Bruijn, what you do is you take the original read fragment and you break it into small sub fragments. Okay, so original, for example, segment was of the length, how much is that? 1, 2, 3, 4, 5, 6, 7, and 8. So that is divided into those even triplets, those three nucleotide size, right? So original fragments are then broken into these short sub fragments, what we call them as k-mers, right? So these k-mers are here, and you just place them, and you again try to solve it with the help of that graph. So that k-mer is, you take the original read fragments and you break it into those short sub fragments or k-mers. So that makes it quicker, right? So this is just a representation. So overlaps, what you do is you can get those original reads, and you go with the overlaps, and then you place them into a shape of a graph, and within that graph then you try to part or you try to get the shortest possible routes. From here you can go here, and after that this fragment comes in, and after that this comes in. So within that graph, within that network actually, suppose these are the flights which are connected all over the world. So you are trying to find the most successful route and the shortest route, and obviously it will have the less cost associated with that. So that thing is one of the method is called as Hamiltonian path, right? Online kilos. Just give me 1 second. I'm sorry, my colleague just showed up to say goodbye. Um, right, so we were here, so that Hamiltonian path and Eulerian path. I'm not going into those details, but these are the approaches, are the algorithms which are running behind those tools like Velvet and Spades and whatever tool. Which tool to use? Obviously, you can see the literature. So people in microbiology or in metagenomics are using, I guess, Spades. In our side, human side, or in the eukaryotes or higher organisms, we go with Velvet. Very interestingly, these tools are named like somebody who liked dresses, maybe velvet and cufflink and top hat and bow tie, and or maybe he was working on some, or he got fascinated by the dresses, right? Or maybe he was a tailor. So OLC based assemblers are Arachne and Cellra and Mugler and Minimal, and these are their names. OLC is the arguably the most successful assembly. The Braun, we already covered that. So original data was, for example, those were your original reads. I think you already got that message. So your KMR size K is equal to 3. So you can start from here THE, and then you can start from second HEA, and then you can start from third, and how many KMRs you can make, right? So that many from this, okay? So a size, this length is, I believe it's 10. So within 10 you are getting 1 2 3 4 5 6 7 8, right? Obviously it will be n minus 2, because when you reach till seventh position, so or eighth position, so obviously you will go out of here, so you just leave the last two characters. So last one is that full segment which you get is FWI. Once you do it from here, so obviously it will be WI. Obviously your KMR size will be violated, so it should be 3. You will get it from rest of them, right? And then you put them together, and finally you get that assembly in different possible ways, and that gives you an overall solution to this problem, right? And here your repetitive regions are also there, which we are addressing in this example. So that's a deep round graph. There is one other thing which you must know about it, the assembly project N50. N50 is a value that tells you the quality of your assembly, right? So N50 is, suppose here you got an assembly that is where you have the contigs, or I guess in this example you will get the scaffolds because you have the ends in between. So original genome is, for example, his size is 200 MB. What is N50? So if the original genome is of 200 MB, what is half of it? Obviously half of 200 is 100. So what you do here is you take the longest scaffold or contig. Again, whichever level you are doing this assembly, you will calculate from there. Okay, if you are stopping your assembly at contig level, so obviously you will take contigs. If it is scaffold, you will go at scaffold levels. So here you get this thing. For example, the length of that contig or scaffold is 70. So first one is 70, and then second one is 25, and you sort them, and then it's 20, and then you have 15, 15, 15, and then you have two of size 10 and 5s, right? So where, like, you can take um, any of these contigs or scaffolds, and you add their lengths. You add their lengths. So whichever set contains the half of the length of the original assembly. So half of the length is 100. So what you do is you take this first one, its length is 70. You add the second one into it, so that makes it 70 plus 25, so that's 95. Still you are short, you are 5 short of the target. So target is half of the length. So how many contigs or scaffolds put together or summed together, their lengths are summed together, how many of them they can cover half of the original genome. So original genome was 200 MB, million base pairs, or MBP. So its half is 100. So when you take the first two, it was 70 plus 25, so that makes it 95. Then you need to take the third one. So if you go beyond 100, that's just fine. That's just fine. Okay. So obviously we need a set where you have three of these contigs or scaffolds, right? And now their length added together, 70 plus 25 plus 20 makes 115. So you have already covered half of it, half of 200. The length of the minimum amongst them. So it's a set containing three contigs. One is of length 70, one is of length 25, one is of length 20. So who amongst them is the shortest or smallest? Obviously this third one. What is its length? So that's 20 MB. So your M50 value is 20 MB, right? I think we have it somewhere here. Length of the shortest contig in a set making up to 50% of the assembly length, right? So most common measure of the assembly quality is the length of the shortest contig in a set. You make a set that makes up to 50% of the assembly length. So 50 total assembly is of 200 MB and 50% is 100. So you make a set. Obviously that set comprises of three of them. Amongst them, this smallest or shortest is of the length 20. So your M50 is here. Right, so let's go back to Linux. Let's go back to some fun. I can start all over. So how can we go to Ubuntu or Linux? What you do? Write Ubuntu here, right? And you just click this thing. So everybody just go to your laptops, right? And open up Ubuntu. And it says root at Wasim. In your case, it might be saying NGS at whatever, Bushra or Sadia or whatever your name is. Right, so you try to do anything like here, so he's not doing anything because nothing is accessible to him or nothing is approachable to him because originally this laptop belongs to a Windows operating system. So Linux is a parallel operating system, so you just need to give him the rights to access its C or D or E drives. So for that the command is cd, so you need to just do this thing /m and you press tab and it will give you the advisory. You can go into media or you can go into MNT. When you go to media, obviously you are accessing your USBs. Here we are not going to the USB, so we do MNT. Then again you press tab, so he's giving you the device that you have C, D and E. And WSL. WSL is actually this Linux, this Ubuntu thing, okay? Alright, so where is my desktop? Obviously on my C drive. So I can go into just one step or maybe I can go into short steps. So I entered into C, now I do ls. So within C, I have users. So I'm a user here, right? So we do cd space users and then within that different users are all users, default user public was in header. Obviously my system is on my name. So then I do ls. When once I reached here, obviously you are seeing that address is being modified here. So right now I'm here, I'm in Wasim header. That's one of the users on C drive, and MNT was where I was giving the access to C and its contents to Ubuntu, right? So Ubuntu accesses this thing through MNT, right? So you do ls here, so it says that here you have my documents, my downloads. Since I made it bigger, so he's showing it in line. So app data and contacts and desktop. I actually wanted to go to my desktop, so where I have my things over there. So I can go like this as well, but I can do clear screen control and cursor up. All right, so where I want to go, cd desktop. Within that folder is NGS, where I put each and everything on the desktop folder, right? So here I asked you to make a folder on your desktops, and that folder should be named as NGS, and within that you have those individual files on which we were supposed to be working, right? So one of them is mu1.txt. So just open it. By default, it opens with this notepad thing. So these are my raw reads. I just want to do their assembly and see from which species they are coming, and that's what I'm doing right now. Okay. So I need to go to NGS. So that's my folder. Can I just take one big jump from here to here, right? Obviously I can do that, or you can take small steps. So you are at the top stair and your legs are stronger. So in our childhood we used to jump from the, I mean not from the top stair to all the bottom, but somewhere where we have 3 or 4 stairs left. uh, as a kid I think everybody jumps, but now we cannot because we need to go stepwise. So I think if you are smart enough, a young kid, let me go back all over. So if I wanna just go all over, I can go like stepwise backwards, or I can go and just in one go CD tilde space tilde, right? So you can go back to your root. Again, you do CD and forward slash and you write M and you press tab and tab will give me the options. You put N also and then press tab and it is written, and within that it's C, and within that it was, what is that, users, and the same header, right, and within that it was, what was that, my top, and within that it was this NGS folder. So I can go in just one step or I can go stepwise, right? Similarly, I can go back stepwise with cd space dot dot. So you will go one directory or one folder backwards, right? But if you put cd space tilde, how do you pronounce this thing? Tilde or tilde or
Tilde.
tilde, right? So my professor, I think was British in origin, Matt Hudson, Nadeem है जिनके पास. So he used to call it tilde, right? Some people called it even tiled, but you can check on the internet what is the pronunciation. Sometimes there is a difference between British and American, right? Yeah, I found uh, American is straightforward, simple. You don't need to worry about the grammar and punctuation and anything, right? So people like us are more cautious about grammars and punctuation and blah blah blah. That's why we are afraid or scared to speak. Just speak, even if you're speaking um, in your own language accent. That's what British used to do when they came here in India. They went to anywhere else in the world. They try to speak the local language. So just fine, right? थोड़ा थोड़ा गोरा सा अंग्रेजी बोलता है, right? So अब भी बोल सकता है. So you just try. So we do ls. So that's our file which we want to do assembly. Which one is that? mu1.txt. You want to see what you have in this mu1.txt. ये lobby क्यों नहीं bypass हो रही, फिर बार बार वो तुड़ंग तुड़ंग हो रही है
पता
बीच
नहीं,
में.
मैंने सबको ऐड तो किए,
क्लास में ऐड करें ना थोड़ा.
क्लास में ऐड किया हमारी टीम्स वही है ना उसमें.
और I think you can set up the meeting rather than starting a meeting at random and sharing the link. That option might correct that. Okay. Right, so that's my file. I want to view it in Linux. Can I do that in Linux? This mu1.txt, I can write it, but obviously he's looking for some command here, right? So that command should be either nano, and then nano is a text editor, so it can file mu1. Obviously, mu1 is has multiple options, so now he got tools. So when I put dot, so he just opened it, so I can open it like that. Control X, go back, control X. Okay. If you are stuck, your command is stuck and it's not getting back, you just kill it. The command is control C or control K, right? You can do that. All right, so where were we? There is another command called as head or mu1.txt, can show us the top few lines, and control L, and when there is a head there should be a tail also. Mu1.txt. How can I repeat the previous command? Up arrow key, okay, and keep on pressing and the previous commands keep on running, right? And these are clear. clear here. I'm lazy, so rather than pressing two keys, I can just go with just one up arrow key, and that will work for me. Alright, so next we go to this document and we can just do assembly today, and probably we can also be able to do the alignment thing. That's what we need to do in a resequencing project or in exomes or genomic variants. If you have the whole genome and you're comparing with the at G38, that's what this first Pakistani genome sequencing project was done. One of our famous scientists from Karachi, from Dr. Iqbal's institute, he um, donated his genome, right? And the PI of that project was Dr. Kamran Nazim. I think Dr. Ishtiyaq was also part of it. And there was another guy, I think he's in Abdul Wali Khan, Dr. Asif. He also worked on that. So that was the wonderful thing that happened. And then obviously our funding agencies, they grabbed their attention. Then we got this lovely center, Jimmy Rahman Center for Genome Research. Obviously we got NGS sequencers all around in the country, right? So here is the command to install that program or that tool or that software built on an algorithm. So obviously it has an algorithm, and that was then translated into code, and that code was then converted into an executable files, and this way you can run it on Ubuntu. So you do sudo apt install Velvet. You don't need to download and install them. Mac users, you just get its .dmg versions and just simply place it there, and you go onto the terminal, and then you can go into that folder and you can run it through their executable files directly, right? So sudo apt install Velvet, I already did that, but if you have not done it, just copy and paste this thing here. Copy and right click. So right click will simply paste this thing here. So I'm getting it again for you. He says that it's already there, so I don't mind, saves my time. Then the next command is, as I said, you just need to give just two commands. Acha ji, so the command is, one of the tools is Velvet H, is the algorithm for hash k-mer generation. It's also called as hashes. The people in programming might know what hashes are. K-mers, right? So K-mers is, we already covered. You take the original read fragment and you break it into short fragments. That k-mer size here is 31. Why not 32? Why not 33? You can try. Okay, but most of the books recommend that k-mer size for Illumina reads, short reads, especially the one coming from Illumina high-seq machines, which are having the length of 100. For them, 31 is the best figure. But there are the tools like Kmer, Jenny, and others which you can use. So you can give your short read data to Kmer, Jenny, or any other tools, and then they draw a distribution, and then they advise you that as per the shape of your data, you would go with 30 or 31 or 32 or whatever. Okay, so you can change that. So Velvet H, and then we wrote a word here that says 31 mer. We are actually converting those short reads into k-mers of size 31, but those k-mers need to be stored in a folder. So you need to give him a folder name here. That is, it's not a command. I wrote 31 mer. You can write my k-mers or Wasim's k-mers, or just k-mers, and a folder named as k-mer is being then created here. That folder does not exist here so far. Okay. All right. Have you seen any folder named as k-mers? No. So it's not there. Right, so we'll just create with this command, and then kmer size is here 31. Dash fastq means the original reads coming here are in fastq format. That means you can also work with the fasta format. So Sanger gives you in the reads or the fragments, sequence fragments in fasta format, so that can be fasta format. Dash short means these are short reads. Now we are getting the longer reads also. So PacBio third generation sequencers or Oxford nanopods are giving us the read in even longer, as how much we saw in that 35 kilobases, the longest amongst
Yes.
them. So that's too much long, right? So here obviously we're doing with short read data. These are the options which you are putting, and I guess they are mandatory, otherwise this command won't run. And this is the input name, the file on which you want to make those gamers, right? So two steps. Number one is make gamers. This is the command. You just copy, you go here and do right click and press enter. So he is making some gamers. So we do ls. So we saw there is a folder here 31 mar, right? आप इसको कुछ और मर भी कह सकते हैं. You can call it some other. So here are your original sequences, right? So it saves the original sequences. You know that quality scores are gone, right? And then it makes another file. Who's Dr. GMS वाले तो नहीं है.
देखो.
Roadmaps you can go and attend outside. Dr. Amar मतलब वही है.
इनके नंबर से दिए.
Right? So he does some calculations अपने लिए कुछ roadmaps. He decides some roadmaps to do that, and log is where he's writing the command and the Velvet variant which I'm using and that record. So that's what he's, and max gamer length, and that's what he is writing something. Logs means logbook. You might have logbooks in your cars, right? So where you're putting when I change the oil and when I did the service and anything. You can check even here. So for example, you do ls and 31 tab. So merge. So that is log and roadmaps and sequences. Well, the second command which we need to run over here is this one. You just go to another within Velvet. Velvet is a software suite. Suite means it has combination of tools. So there is another tool called as Velvet G. So Velvet G 31 mar उधर जाके मरे, right? So we are asking Velvet G, right? We're calling the tool Velvet G, right click, and just showing him the folder where they have those 31 mers, right? Where those k-mers are there, right? And it will have the raw materials. Okay, the example which I gave to that workshop people, right? So bride's family is here, लड़के वाले आए हुए हैं. What mothers do is they do half cooking and put it in kitchen, and they tell the girl go there and just put the things together and make a biryani out of it and bring it, right? So that's what we are asking this tool to do. So Velvet H is creating gamers, and now Velvet G is having those raw materials. He will simply go there and perform that assembly thing. It will be real quick. Obviously we have just 1000 reads here, but in real life you might have 10 million 10 MB data. So it will take some time. Okay, so it's not that simple. नहीं अपना देखें, आप अपना मोबाइल देखें.
नहीं मैंने मोबाइल से किया वाला.
अच्छा right. Okay, so what happened over here? So now we also see what is there in that 30 mark thing. We see that we get pre-graph and last graph and sequences and contig.sfa and stats.txt. You can go into those individual files and check what's happening here. So some of the files, more files are created here, right? So we can classify them on the basis of their types. So contigs.fa is a FASTA file where you get the contigs. Stats is he's doing some hisab kitab or statistics. So longest contig and shortest contig or length of the contig here. The contig number 1 is of length 155, and then you get, so anything beyond 31 is a fair deal because your gamer was of size 31, so anything beyond 32. But obviously your original read fragment was 100 bases, so your smallest contig should be even greater than that. But normally what we do is we take the top 300 or 400 or 500 contigs and discuss what they contain in them. So we can just open up that file for us. So that's the contig file, right? And that is where you see this first contig is of 155, and whose length was the highest. What I can do is maybe I can go here and open up this thing with open with right click and open with, and I will try to open it with Microsoft Excel. So go into Office and Office 16. And another thing which I can do is I can open up Microsoft Excel. Into use sorting in Excel, and I can do it. All right. So new, open, and then from here I can go to this PC and I can go to browse, and that's there on desktop, and that's there on MGS, and within that 31 more thing. I can go with all files, stats, and open, and let's see if it opens. So my data has headers. So delimiter I'm using here is okay. Next, yeah, finish. Perfect. So I can go into data and sort, and within sort I can sort on length, and on length largest to smallest. So 535 length ka that contact is there. ID 11, right? So ID 11 is 535. That's good one. Where is 530? I can do here 5. This one, yeah. So we got it. So maybe you can take that big one and you can put it into Blast or any other tool and see which species it was. Are you guys familiar with Blast and Blast? You know, that is the wonderful that happened in our field. So Alstul and his, you know, colleagues or his students maybe at all, right? So they have done a wonderful service to our community. So you put it as, and we just go with Blast N. The purpose was not to teach you Blast, just to look into what you have in here. Or you can go to the Blast and you can upload all those context and see what these context have in them, right? So yeah, so that is the Arabidopsis thaliana 81G299330 messenger RNA complete coding sequence. That's the best hit, 99.12% identity. So that's coming from Arabidopsis thaliana. Obviously, it is this sequence which we actually get it from our, you know, experimental data sets. Over there I had Arabidopsis and soybean, and then over here we work with humans, and we work with bacteria, and we work with fungus and everything, right? So that's what you got. Okay. Even you can take this context.fa thing and you can go to Blast. All right, and what you can do over here is, oh, I'm sorry, again you can upload the whole file rather than, you know, picking up on these context things and go here. So what BLAST does is, BLAST stands for Basic Local Alignment Search Tool. A local alignment is if you are comparing two or more sequences with each other, so it tries to find you know, the patches or the regions uh, over those sequences where they have the maximum similarity or identity. What is the difference between identity and similarity? Identity is T versus T. Similarity is, you know, on one side it's A, but on the other side it's G. So both are purines. So that's, they are somewhat similar to each other. In amino acids, for example, two amino acids are not matching with each other, but they are coming from the same group. Like both are covalent or both are, you know,
Glutamate and aspartate.
yeah, something like that. Exactly. All right, so what you do is choose file and So in real life, for annotation, we are not actually going with BLAST. This is just to give you, since our data set is small, we use Augustus, and what we used for after spades, what she used in bacteria, quast. Yeah, quast. Oh, by the way, there is one exercise in Galaxy. So we get those reads all over. In Galaxy, what happens is you can go to a website and you select the species you want to do this analysis. So automatically it's read data from SRA, short read archive, where you have that read data in CBI. So that is just, where are those? So that is automatically goes to Galaxy and you can analyze that. So please finish those exercises yourself. We're not going into Galaxy, but
Excuse
because
me,
that,
sir.
yes, please.
Sir, is that we have divided the data we are provided into 31 sets of sequences, right?
Uh, 231. So we got 231 contigs
No,
here.
31, 31, 31. So isn't
31.
it that the sequencing data already has certain amount of sequences? How can we divide or decide that we have to convert the data into 31 MERS or 20 MERS?
Oh yeah. So that is again depending upon which species you are working with. There are the guidelines people have tested, right? Or there are the tools also which you can use them, or you can trust on those research papers, right? So mostly people are picking this 31, this Kmer length of 31. Okay, all right.
Sir,
So
if we take like half the length and
so
then see the
50,
shortest one, that will be N50 value of this quantity, right?
uh, N50 is something which is here. So that is once you get your assembly. So for example, our contigs here were notepad कहाँ गया? Let me open that thing again. So that's their lengths, for example, right? So suppose so total assembly length which we covered so far is, for example, 500 or 600 or something. What is the length? It should be maybe 1 kb or something, or 2 kilo bases. So what is the half of 2 kb is obviously 1 kb. So how many contigs you just sort them and then you sum them up if that number, and obviously you need to attach this third one also. So these three they sum together makes, they should cross 1000 but they should not be less than 1000. The total length of that
Then
is,
N50 would be 234, right?
yes, exactly, the shortest amongst them. So that will be your 250. So obviously those tools they also report them. But here we are not going into that much detail. So, and once I guess in Velvet G, I guess there is a flag here, right? So you can get the N50 as well. So in that case, you can get the whole output in which they also report you uh, the mm, nice no hub where it gives you the statistics also. Uh, Nice means be nice on resources, no hub maybe, depending output to no hub dot output. So yeah, so he is just, oh yeah, so since we had all those things which are assembled here, so he just ignored that. But if we see what he has written into this no hub file, so I think he gives us this statistics in a file. Right, so that is where he is just writing all those steps rather than printing them on console. Yeah, yeah. Final graph has 44 nodes and N50 of 109. Maximum is 534. Total is 4481, right? So max length is 534, right? And total assembled region is 4481. That is obviously half of it will be, so amongst them, I guess he stopped at 109. That was the shortest amongst them. using 0 out of 100. So that output was down there. So final graph has, I think it should be there in that stats thing also. Let me find it. Just look into those files. Maybe in log maybe. It's very interesting question. Yeah, so it's there also. Okay, excellent. All right, so you want to go more, or I think we should stop here till assembly things. Should we cover the alignments or mapping? Oh, by the way, I've also installed this tool called as tablet. So over there you can open up an assembly also. How can I go to my desktop? So I can go here and here. So firstly, where is my assembled? Yeah, so within that, context, sequences, stats, formats, maybe context, open. I can open up the some of the test examples are also here. I'm sure how to get started. Open an example assembly. So that is the context. Then I guess I need to give the original reads also. ROS 454 original is 31 compressed with gzip to 5 MB. Right. So context plus ACE assembly file. Maybe I can go with something like a small data sets. Just want to view. Right. So yeah, so here is a region that has been assembled. The reads which are aligned here, that many, right? And each position is then, I mean, the place where which are jam packed or crowded are the ones which are highly covered, right? And here we are getting that consensus thing. So consensus which we constructed from here, it's a G here. All reads are supporting that point that it's a G here. These are all A's. At some places we might also get, for example, some of the nuclear some of the reads which are not agreeing to, like this one originally was C, but some amongst them are saying it's a G here, but obviously that call is not that higher. So some of the places might have some regions, so that is, I guess, this place where you can see one mismatch. I think this is the place where we can see there is the assembly quality may not be that good, but mostly the reads are supporting that. So the individual reads, you can just zoom in and see what they have in here, right? So that's the viewer, alignment viewer. So one of them is this tool. That cloud is just number of reads mapping to those regions. The regions where you have less reads are obviously having low coverage, and the regions right. So that is obviously this region. So some of the regions are good covered, some of the regions are not that good, right? So you can see that. All right. So how much time? 20 minutes. Maybe I should cover that alignment part also, if you guys are okay with that. Now, content copy. Sadia wants me to stay. I can take your questions, please. Just practice that over the weekend. Any issues, weekdays even, when you'll be free from your exams.
Right.
So she can take your queries on Fridays.
I can take it from that.
No, just prepare well for your exams. You need to maintain a good grade, right?
Sir, can
Anyways,
you use it for long reads also?
sorry.
Or is there a limit?
Sorry, say it again.
Sir, can we use tablet for long reads also? We can also visualize
Yeah, yeah,
long reads in
yeah.
after nanosequencing.
Nanopore, you got the nanopore data?
Yes, if we get nanopore data, then we can use tablet.
I believe yes, because but the things were under development for nanopore and rest of the technologies, but most of the tools are somewhat similar. There is this UCSC genome browser, but that is a little bit, cumbersome. It takes more time and steps to configure that. We can do it, but tablet is really handy and real quick, right? All what you need to do is you need to give him the data into that particular format. I just need to check for my own assembly, which I did that. I think, yeah, so you need BAM file and you need FASTA file. So that's why he was, yeah, so original FASTA file and then BAM file. That's what he was taking, right? So maybe I can try, you know, the original reads and this assembly thing. Probably it might gonna work with my data sets, the original one. I just wanted to see um, how are, so that's where I can get the sequences. Maybe they are in FASTA format. No, but he disassembled them. Maybe I can check later on and then figure out how to do that. For the alignment thing, you need to just give him the BAM file, SAM file, and the original FASTA reference on which you are aligning, so it works. All right. So anything else? So today we covered the assembly part. Next week we're gonna go with that variant calling thing, and then we also go um, with the with the differential gene expression. All right.
Excuse me,
So,
sir.
Gigi.
Sir, when we have the data like whole genome data or whole exome data of humans, so do we use the same procedure for alignment because the data is very extensive, or do we have other softwares for the purpose?
There are so many, but these are the most commonly used: Bowtie, BWA, Velvet. As I said, spades we used for microbial data sets. So the best approach is we can see what most of the community is using, right? Um, Even we are using Velvet, but spades is doing similar. So you just need to download the tool, and there are the commands which are there in the manuals, so we just need to practice and follow them, right? But
Yes, sir.
those commands are just similar, so I'm sure you would figure it out. Okay, any problems, we are here to help and assist. So my students, they have explored so many tools. Probably I did not go beyond myself in while during my PhD. I went with Bowtie, and I went mostly with R because my data was all about RNA sequencing. While we were doing exomes, what you were using
For the exomes,
for the exomes?
alignment, BWA,
BWA. So Sadia was using BWA followed by
free base,
yeah, yeah.
and then the NOR.
Yeah, we will cover that part also because I guess most of you are working with exomes, right? And obviously the metagenomics. I will cover the basics, but if you want to go to the advanced levels, obviously we can make use of experience of our students also. All right. Anything else? Are we good to go? Would be nice.
Yep.
Yes. Thank you so much. Okay. Thank you so much. All the best. See you next week. Best wishes for your exams, our own students, including Sadia and
Yes,
Hoshar.
sir, we need that.
Welcome.